1. Found in plant cells, prokaryotes, fungi, and some protists, not in animal cells. 2. Function: protects cell, maintains shape, prevents excessive water uptake. 3. Plant cell wall, cellulose. Prokaryotes, peptidoglycan.
Fungi, plant cell wall. 1. Extracellular structure. 2. Secreted by cell.
3. Porous, allows water and dissolved material passage. 4. 3 main layers.
Primary cell wall, PC. Middle lamella. Secondary cell wall, SC. Primary cell wall. 1. Develops during cell. 2. Thin, flexible. 3. Cellulose microfibrils.
Hemicellulose. 4. Adapted for growth. Secondary cell.
1. Found in sclerenchyma. 2. Thick, rigid. 3. Cellulose. Hemicellulose. Inorganic source. 4. Provides mechanical support, definite.
Middle lamella. 1. Between primary cell walls of adjacent cells.
2. Holds cells together. 3. Magnesium and calcium salts. Plasma membrane. 1. Also called cell membrane. 2. Boundary of protoplasm and most organelles. 3. Found in all living prokaryotic and eukaryotic cells. 4. Controls passage of materials into and out of the cell. Composition. 1. Proteins, 60 to 80%. 2. Lipids, 20 to 40%. 3. Carbohydrates, small quantity. Fluid mosaic. 1. Phospholipid bilayer with embedded protein. 2. Proteins scattered throughout membrane. 3. Membrane is 7 nanometers thick. 4. Lipid bilayer. Hydrophobic ends face each other, hydrophilic ends on surface. 1. Asymmetrical membrane. 2 surfaces and halves not identical. 2. Proteins drift sideways in fluid bilayer. 3. Carbohydrates attached to proteins, glycoproteins, or lipids, glycolipid. 4. Filaments of cytoskeleton support membrane. Functions. 1. Controls transport of materials. 2. Provides mechanical support. 3. Regulates cells' interaction with environment. 4. Maintains pH and ionic concentration. Protein function. 1. Transport channels, carriers. 2. As 3. As receptors. 4. As antigens. Glycolipids and glycoprotein. 1. Act as cell surface. 2. Involved in cell-to-cell recognition. 3. Stick correct cells together in tissues. Membrane fluidity. 1. Controlled by lipid composition. 2. Unsaturated fatty acids increase. 3. Cholesterol stabilizes lipid by. Study techniques. 1. Atomic force microscopy. 2. Super resolution fluorescent microscopy. 3. Scanning electron microscopy. Thank you. Like and subscribe my channel.